Good evening, everyone, and you're welcome back to the channel. This is the second part in a three-part series where we are discussing the structure of the bacterial cell. This time around, we'll be looking at the cell wall, the bacterial cell wall. And many a times, we must have heard that the bacterial cell is called a peptidoglycan cell wall. The peptidoglycan cell wall, and this is important because this is the structure that the bacterial cell wall is made up of. Now, what does the bacterial cell wall do, and why is it important? The bacterial cell wall helps to give the bacteria strength and rigidity, keeps the bacteria firm, keeps its structure strong so that the bacteria is able to withstand all that the environment throws at it. Now, the bacterial cell wall is very, very important because in the event that the bacteria gets thrown into a hypotonic solution, that is a solution that has high water content, that bacteria is able to survive because of the strength of the bacterial cell wall. The cell wall is strong enough and does not allow the bacteria to burst and leak all its substances or internal materials out of the cell. This is very important. It's very, very important, and it helps the bacteria to be strong. Now, the cell wall has quite a number of functions. Also, it helps to keep the shape of the bacterial cell. The shape of the bacterial cell is very important. We've discussed some shapes of bacterial cells, and these shapes range from oval to spherical to all these different shapes are based on the shape which the bacterial cell wall gives to the bacteria. This shape helps to give the bacteria a shape. So this is part of the functions of the bacterial cell wall. Now the question will be, this bacterial cell wall, what is it made up of and what does it do? The major structure that we have in the bacterial cell wall is a structure called peptidoglycan. Another name for peptidoglycan is murine or peptidoglycan. Peptidoglycan or murine is the important structure that we have in the bacterial cell wall. Now the cell wall also functions for bacterial growth. If bacteria is going to grow, the cell wall is one of the areas that need to divide for a parent cell to become two daughter cells. This is very, very important for the bacterial cell. Now, like I said, the bacterial cell is made up of peptidoglycan. Peptidoglycan is a material that is not porous. It's impenetrable to water and doesn't allow water to easily flow through it. It's also selectively permeable. It sometimes allows some substances to pass through and others do not pass through. This is important, and it is made up of a cross-linked polymer. This cross-linked polymer is what we call the peptidoglycan. Now, this peptidoglycan, I've explained before that you need to understand the concept of monomers and polymers. Polymers are giant molecules and they are made from monomer units. When you couple monomer units together, you make giant polymer molecules. Now, the cell wall, like I said before, is made up of a peptidoglycan layer. And this peptidoglycan layer is made up of monomer units, a lot of monomer units. Some of the most important monomers that make up the peptidoglycan cell is one, N-acetylmuramic acid. N-acetylmuramic acid. Another monomer unit is N-acetylglucosamine. So N-acetylmuramic acid, N-acetylglucosamine are all structures that you can find in the bacterial cell. We also have some proteins in the bacterial cell, which include D-alanine and L-alanine. D-alanine and L-alanine. We also have D-glutamate and adiamino acid. All these six monomer units that I've discussed are the monomer units that come together to make that layer, that structure that we refer to as the peptidoglycan layer, and it's very, very important. It's also important to note that as microbiology students, we are still going to have a video where we'll discuss gram staining, the procedure and the microbiology and science behind gram staining. You find out that in gram staining, you have the division of bacterial cells into gram-positive and gram-negative bacteria. Now, a lot of bacteria that are divided into gram-positive and gram-negative are divided on the basis of the structure of their cell wall. We found out that the gram-positive bacteria have a much thicker cell wall than the gram-negative bacteria, so they're able to retain more of the stain. More of that will come up in our video on gram staining. This is important to know. Gram staining procedure is very important, and it shows the difference in the cell wall structure of different bacteria. This is important to note. Now, after the cell wall, we have an inner membrane, which we call a cell membrane, that is present in the bacteria. This cell membrane is made up of a phospholipid bilayer. That is, we have two layers, and these two layers each layer has what you call a hydrophilic, that is water-loving phosphate head, and a glycerol tail. A glycerol tail that is hydrophobic. That glycerol tail does not love water. So you have two layers like that, a phosphate head that is hydrophilic and a glycerol tail that is hydrophobic. This is important to note, and this is what we have in the cell membrane of the bacteria, which also has a cell wall. This is important to note, and I hope we have been able to understand what the structure of the bacterial cell wall looks like. Thank you for listening to this video. Have a wonderful day.